results we've been getting with these. But anyway, where do we get them? Well, we can get them right now from 11 different species of mammals. Mice, rats, rabbits, cats, dogs, goats, sheep, pigs, horses, because that's an age, cows, and humans, bovine. Equine bovine. So 11 different species, we can repeat the results, not a problem. We use sexually mature because they're the easiest to get our hands on, but you can go newborn and you can go geriatric. And our oldest geriatric was an 87-year-old female that was actually a diabetic, type 1 diabetic. And they amputated her leg and we got the bottom part. And we isolated the stem cells from that. And we isolated stem cells from the 67-year-old male. So those are our oldest so far as humans are concerned. Mice, where we are here, NIH has an aging model. And it's called CBF1 mouse. And most mice live to the ripe old age of about 24 months, maybe. These mice live to the ripe old age of about 30 years of age, which is equivalent to about, not 30 years, 30 months, which is about equivalent to 120 days. Wow, man, that's an old man. Wow. 30 months of age, which is equivalent to about 120 years in human, and we can find them there, so full range, no problem. They don't decrease with the other one thing I wanted to tell you is progenitor cells, you hear about the thing about stem cells decrease with age of the individual. That's exactly right for progenitor cells, the ones that are committed to particular cell types. But our stem cells, the ones that aren't committed, they just go on and on forever. I mean, they don't decrease with age. We've tried some experiments to prove that, but what we found was that the newborn ones, it takes us 15 minutes to get them out of the tissue. The geriatric ones, it takes us 8 hours to enzymatically digest them out of the tissue. So they're more tightly bound in the tissue in the old farts rather than the young farts, so to speak. But that's the only difference. But they're still there. The next question is, where can you find them? The answer is anywhere. They're present everywhere. Everybody says, oh, they're only present in bone marrow. Nope, sorry, afraid not. Our gold standard was skeletal muscle because originally that's what we were isolating to get the myoblasts. And that's the one we normally went for. Recently we found them in blood, which is even nicer. So we started out with doing punch muscle biopsies to get them out of humans. And people say, no, they don't want to do that. They'd rather have them out of their hip instead of out of muscle. Anybody in here ever gone through a bone marrow transplant? That ain't the word for it. For the recipient, it's great. For the donor, it's hell. You're in the hospital, they put you under general anesthetic, they withdraw the bone marrow, you're in the hospital the second day trying to get over it, and then you're sore for about 2 hours, bone marrow donor. You're sore for about 2 weeks afterwards, and stuff like that. For the recipient, they accept it like a blood transfusion. The donors, what the problem is. So, but people would say, no, I wouldn't want skeletal muscle. So we tried dermal biopsies, just do a dermal punch. We could drive stem cells, not a problem. No, you have a little scar, don't want that. Then we found out we could get them out by venipuncture. All we have to do is just withdraw a little bit of blood. We got the stem cells. So that's what we're doing right now. That's why this thing starts. So now this has become our gold standard rather than skeletal muscle. Skeletal muscle takes us about 2 days of very intensive work to get the stem cells out. If we use blood, it takes us 2 days. Stick them. 2 days later, take them out. They're separated. I mean, it's just easy as pie. No sweat. I go for things that are easy. So anyway, we have right now, we have isolated out blastomere-like stem cells, which are totipotent, pluripotent, which we call, instead of saying blastomere-like stem cells, we call them BLSCs. Pluripotent are ELSCs. We call them according to, they're like epiblast cells, the inner cell mass, ELSCs. Our GLS mesodermal stem cells here, and we can also isolate those. And we clone them from single cells. So we've taken one cell, grown it up, applied our inductive factors to it, and our BLSCs will form 65. There's 220 different cell types in the body. We have assays for 65 of them. One cell of a totipotent stem cell will form all 65 different cell types. Of the pluripotent ones, they'll only form 63. they won't form a totipotent stem cell, and they won't form a germ cell. So it's only 63 out of 65, whatever. My peers in the literature, we clone from a single cell. One peer, Catherine Verfey, clones from 10 cells. She starts with a starting population of 10 cells and gets 3 cell types. And she says she has pluripotent stem cells. Don't scratch my head here, whatever. And Osiris, which is a company that has mesodermal stem cells, they're in the news right now about their cells can immunoprotect people. They started with a population of 4 cells and say they have a clone that will form 3 cell types. Still scratch my head, whatever, you know. We do things differently. So, here are pictures of my cells. Here's the star-like ELSC. It's about the same size as a red blood cell. Here's our little blastomeric-like stem cells down here. This one is in prophase, for any of you that have taken basic biology here. This one's in metaphase. You can actually see the chromosomes lined up along the metaphase plate. So they are dividing. And we have 2 of them, that's why I had that there. And they range anywhere from 0.2 microns, which is unheard of for a cell, up to about 2 microns, which is still unheard of for a cell. It's too small. Cells can't be that small. They aren't. So we grow them up, separate them. We separate them by cell sorting, different techniques. And we can get individualized cell populations of ELSCs. Their marker is called stage-specific embryonic antigen. That's the name of the antibody that we use. In BLSCs, they are carcinoembryonic antigen. Now that's an interesting name, carcinoembryonic antigen. It was named because they found adult cells, embryonic-like cells in an adult. Heaven forbid, it must be cancer. It must be an embryonic cancer. So that's how the antibody was named, but actually it lights up these stem cells. And like I said, these are isolated, they're separated. So our ELSCs, 6 to 8 microns in size, they cryopreserve at -80. They're not contact inhibited, meaning they'll form multiple layers in culture. They're normally quiescent, like my daughter sitting on the couch. You don't tell her to do anything, she just sits there, nice and quiescent. You know. Rat, we have about 300 population double in human, we have one out 400. The reason is they're not out to 690 because we haven't had them long enough, this sort of thing. They'll form everything. Same thing the embryonics will do, except they won't form germ cells, so we call it a pluripotent stem cell. And we have our blastomeric-like stem cell, less than 2 microns, freezes at the same temperature, multiple layers, quiescent, extended. We have a rat out, approximately 300 again. We've had these cells about 4 years now. We've known about them about 4 years now. They'll form spermatogonia, so we call this cell a totipotent stem cell. Same as the embryonic. So we have adult stem cells that I can isolate from any one of you that are equivalent to the embryonic stem cells. Just keep that in mind. So here's this. And a lot of people, if you read the literature, say, oh, it's actually de-differentiation. You have adult cells that go back up. Doesn't work that way. They always go down. They always differentiate from least differentiated to most differentiated. The ones where they have said they went back up the other way, their cultures were contaminated. Because we tested it. We used clones of the little guys and put them in with clones of the differentiated ones with different markers, let them grow several passages. Lo and behold, we had, say, a muscle-derived progenitor cell. It died off and we got neurons. Unfortunately, it had all the markers of these guys, it had none of the markers of those. But again, it's real small cells, so it's hard to miss. So now that you know that stem cells, what can stem cells do for you? What do you think? Well, the example I brought was type 1 diabetes. Okay? We have them in a lot of different systems. I have them in myocardial infarction. I have them in Parkinson's disease. A lot of these are this is my, the yanker, I should say. Okay. Type 1 diabetes is a failure of traditional therapy with exogenous insulin to cure the disease. It won't cure it. You have complications, many complications. It can lead to deaths, cost of treatment skyrocket. Over 16 million Americans suffer from the disease. Annual health care costs exceed $92 billion. Now somebody would ask me, well, how can I treat type 2 diabetes? And my answer would be diet and exercise. But for type 1, you need something special. And you need more islets, basically. Now, Edmonton, Canada, about 4 years ago, came out with a protocol. Instead of transplanting the entire pancreas, what they did was isolate individual islets out of the pancreas. And then they would shoot them into the portal vein, which goes into the liver. Those things would get stuck in the liver, and about 75% of the people would survive the operation. And of those, 75% would survive, otherwise you'd have liver function and people would die. But the ones that survive are out 4 years now, exogenous insulin free. So the technique works. We're still trying to, for 75%, yeah, it works. There's still that 25% where it doesn't work, but 75%.
They died from the procedure, or
They died from the procedure.
From the procedure. So they volunteered with type 1 diabetes.
Yes.
Which they could have managed with medication.
Well, they were to the point of being brittle diabetics. And at that point, you can't manage it with insulin.
Okay.
So it was, they're on their last whatever.
Okay.
So the islets they used were too large. They clogged the vasculature in the liver, and basically the people died. Okay, that's one of those things. But the 75% that did survive, they're out 4 years. So, hopefully you're one of the 75%. You know what I'm saying? Okay, so anyway, they take a pancreas and use enzymes on it. They try to get out these little guys, which are the islets here, and then transplant them. And actually, there are more islets in the pellet than there is in this fraction right here, but there's a lot of trashy material, so they just toss that.
excuse me,
Yeah.
the pancreas that they use to retrieve the
Cadaverous.
Okay.
The person has to die to
Right. Okay.
donate the pancreas. Yeah. That is a problem. Okay. Anyway, so promising experimental cure. Obstacle. Now the other problem is it requires 2 people to die to get enough islets to save 1 person.
You could, well, with replication of the islets through
They have the biological clock, they'll croak. So you need 2 people. So, and there's not enough donors anyway, for regular organs, let alone pancreas organs. So the call went out for, is there any way that we can generate more tissue? Need alternative sources of insulin producing islets. Now, where are we going to obtain those particular insulin secreting cells? Well, 3 groups popped up and rose their hand. One group was pancreatic progenitor cells. This is Gordon Weir's group at Harvard. You have embryonic stem cell people, and there are more places. And then there are adult stem cell people like myself. So we all raised our hands and said, we can do this. Well, so we have pancreatic progenitor cells, like I said, Gordon Weir's group in Harvard. And they found that if they took 50 pancreases, they could produce enough beta cells for single islet transplant. 50. 50. Kind of going the wrong direction as far as I'm concerned, but whatever. They'll get to it eventually, maybe. Okay. So now we have the embryonic stem cell people, and they say, well, we have potential, or stem cells have potential unlimited source for proliferation. So we could proliferate ad nauseum and get you all the beta cells you need. Okay? And that's basically what they did. And so we're going to generate a lot of cells. Then we're going to allow spontaneous differentiation. We'll harvest only the insulin-secreting beta cells. So there are a lot of cells we're just going to trash in the process. And then what they did was test for functional activity. And to do that, you do what's called a glucose challenge test. And the lady that did it, Nadia Lumelski out of NIH, she did it, what I would say correctly. And the way she did it, the way she did it, start off with your embryonic bodies, uncontrolled differentiation, spontaneous differentiation, isolated only the beta cells, harvested those, and tested them and used the same pot of cells for the test, which makes perfect sense. I mean, most people take 3 different pots of cells to make the test. You're only one person, so you're essentially one pot. So this is normal glucose level, and then they change that out for 1 hour, and then maybe a 25 millimolar, and that's the actual test. And her problem was she used a non-specific insulin RIA, which means we put bovine insulin in the media, and her insulin RIA would measure it. Doug Melton's group at Harvard said she did it wrong, she should have done it, yada, yada, yada. Their results are exactly the same, whether they used an insulin-specific one or not, so I just keep hers on there because she was the first one to publish. But anyway, what she showed was that it would secrete 5% of what native islets would secrete. Okay, by her procedure. So, generated 5% of insulin compared to native islets, and that's nanograms insulin per nanogram DNA generated. That's just standard amount. So her suggestion was you just implant 20 times more embryonic beta cells. No problem there. Okay? So they have beta cells, it'll produce insulin, it's just 20 times more. So you just expand the operation. And there's me, in my group. We have potential for unlimited proliferation as well. Well, we have some problems in that our cells don't spontaneously differentiate. Big problem here. So we had to devise the cocktails to get them down the lineage, which we did. So we took adult, and since I knew both of these would form insulin-secreting cells, I used both of them. Just kind of making sure my bets were covered. We used a chemical-controlled stepwise induction. We induced pancreatic progenitor cells. Then we used Gordon-Weir's procedure, the pancreatic progenitor people, and induced them into three-dimensional structures that actually contain beta cells, alpha cells for glucagon, and delta cells for somatostatin. So we actually created islets, not just beta cells. And then we tested for functional glucose activity. And I'll show you what we did here. So here's a little study. We went from BLSCs and ELSCs, we went to endos, and then we went to progenitor cells, and then we used Weir's cocktail and got them down to 3D islets. And the pictures are, this is endo-SCs. We brought them down here. This is antibody for insulin, and you can see the dark stained insulin cells here, and they're just kind of diffuse. And this is glucagon diffuse, and this is somatostatin. And then we formed three-dimensional structures. Structure actually goes out there, and the structure is right there. That's it. So we knew that we could do it. So we repeated it. And I took this picture, kind of looked like a little dog, you know, tail and the leg and stuff. Okay. This is normal islets, a combination of islets, single islet, and this thing. And this is what we tested against those guys. Okay? And we did it the same way. We did the same pot of cells. One pot of cells, tested all three ways, just like Lemelski. The difference was, since we were using rat cells, we used a rat-specific insulin RIA. We tested against bovine insulin, we tested against human insulin, we tested against porcine insulin, and it would only measure rat insulin. So we knew that what we were measuring specifically came from the rat cells. It didn't come from anywhere else. No ifs, ands, or buts about that. Okay? We took native islets from Worcester-Firth rats, so we took rat cells there. We induced our little doggy thing here. We also tested pancreatic progenitor cells, since we had that diffuse population, and we compared them to Lemelski's work. And that's what we got. This is normal. These are the three-dimensional structures here, so a quarter to a half, and then the blue was our diffuse cells, which was a half to basically 100%. And here's Lemelski's work at 5% across the board. Still in nanograms, insulin per nanogram DNA. So we would say, okay, you implant more than one cell type. You implant both the 3D islets and the induced pancreatic progenitor cells. So we went to the FDA with that idea, and they said, no, can't do that, because you induced with animal proteins. Okay? You can't use animal proteins with human cells, because you might have an animal protein attached to that cell that the person is allergic to. Can't do it that way. So kind of went off on the back burner. We did some other experiments and we found that we could co-culture our BLSCs and ELSCs with adult tissues and they would form that adult tissue. Now that's interesting. Okay. So what we did, so anyway, this was, okay, promising cures, 50 pancreases, more of this, and double this one. Okay, but we really can't do this one because we can't use animal proteins.
Dr. Young, how do you get away with, say, when you use a pig valve for a heart transplant with the proteins?
Because the pig has already been approved by the FDA.
Oh,
But see, we were using bovine and mouse, and we were just using cocktails that we came, just, you know, kitchen sink stuff that we could come up with to induce these things down the line.
Okay.
And then the FDA wanted to know what we used, and we said we use cocktails of animal proteins. And they said, no, you cannot do that. You have to use specific human recombinant proteins if you're going to do it. And that costs a lot of money, and my lab operates on a shoestring, so we kind of backpedaled there. But now, is this the best Dr. Young's lab can do? Guess what, guys? No, it's not. Especially since we know that we can co-culture now our BLSCs and our ELSCs with adult tissues. So we can make islets by co-culturing with islets. Because islets secrete a factor that forms makes progenitor cells more islets, so we just put our stem cells in and they do the same thing. But we wanted to do it something, you know, even more exotic, is we wanted to create our own organs. So you could transplant in an organ, so instead of trying to, you know, lose that 25% population, if you have an organ that you can transplant in, if anything goes bad, you can take it back out again. If you shoot islets into a liver, if something goes bad, you got to have a liver transplant or you're dead, you know. So we're going to try to create an organ. So to create an organ, we take a native pancreatic matrix, and actually we use pigs since they're already approved for FDA, and we took all the cells out of them. And we started with 20 different detergents, and I mean we were using Dawn, and we were using household liquid stuff, just anything we could find detergent-wise. Tried all different combinations. We started with 20 different ones. The standard ones from the chemical lab, and the other non-standard ones from my wife's sink in the grocery store, and anything we could find. Narrowed it down to about 7, and then we started combinations. And we found 2 combinations that worked. And then what we did was to add the BLSCs, and then we added the donor islets. And I'll show you how we did this. So we did the decellularization, took all the cells out. So in our matrix A, there's actually decellularization agent A. You have some type 1 collagen bundles, and all these little strings are glycoproteins. And if you notice, there's more glycoproteins in B than there are in A. And that does make a difference, and you'll see that later on. So then we layered our cells on top. And these were rat cells. So we layered, and we mixed the culture. So all these little bitty spots that you see are BLSCs, and the bigger ones are ELSCs. So we just blanketed that matrix with cells.
So these were rat cells on top of a pig
Exactly.
pancreas.
That's right. So we put rat cells here on top of the pig matrix because we wanted to be sure that what we're measuring was actually rat insulin, not pig. Because we had to try different combinations of things. And then we put donor islets, also from a rat, on top of the matrix. And lo and behold, this is what we started with. See the small size here. And this is what we ended up with. So the larger size is here. So we grew bigger islets. Then we tested. Now we had expected nanogram per nanogram insulin DNA, you know, close to normal and this sort of thing. It's not what we got. So anyway, this is this. So we did that. This is what we got. 250 times the amount of insulin that a normal pancreatic islet would produce, our combinatorial structure would produce. And it's micrograms insulin per nanogram DNA. So we standardized the amount of cells present, but because we have this combinatorial matrix with the cells, we could produce 250 times as much insulin. So here's our artificial pancreatic organoid, as I call it right now. So we have growth violence on these cellularized matrices, provides good culture environment, induced progenitor cells, forming 3 little deals here, matrix with 3 LCs, ELSCs plus islets, more intense insulin response than islets by themselves, potentially transplant significant. We figure we can take one cadaver pancreas and transplant 50 people. So 1 to 50 odds is better than 50 to 1 odds in this sort of thing. So that's where we are right there with those studies. Now, if you want to alleviate donor shortages, you got basically, in my mind, 2 choices. Embryonic stem cells or adult stem cells. Now, Tony Adela, his group, about a year ago now, published on amniotic stem cells, meaning stem cells that you get from the amniotic fluid of fetuses. And yes, they are pluripotent. He's found pluripotent cells. He hasn't found totipotents yet, but I'm sure he will shortly. But they're still allogeneic. They're still not self. And that's what the key is anyway. But anyway, embryonic stem cells, if you want proliferation, both of them proliferate out of style. Unlimited proliferation potential. Not a problem. Both will form all cell types of the body. No sweat. Naive in vitro. These will spontaneously differentiate if you don't put inhibitors with them. These will sit there and do absolutely nothing with or without inhibitors. It doesn't matter. If you put them naive in vivo, you're going to form cancer cells. Okay? If you put adult stem cells, they'll either remain quiescent or they'll track to the damaged tissue and restore the organ. And we show that in heart, we show that in Parkinson's disease. Those are our 2 in vivo models we've shown it in. And we're working in some human models right now as well. If you implant in vivo, you have to implant differentiated cells if you use embryonic stem cells. You don't have a choice, otherwise you're going to form cancer cells. Our best results are the most naive cells. The GLSCs work nice. The pluripotent cells, stem cells work even better. And the totipotent stem cells, or the VLSCs, work the best. Because basically what you're doing is giving the body building blocks for it to use to make what it wants, it needs to use to repair the tissue. And case in point, one of my collaborators is working on a Parkinson's patient. And they're giving a compound that pushes VLSCs out of the periphery, and he was going downhill precipitously. He bottomed out, so he's now stabilized. But added some more, and he's still stabilized. They didn't know what was going on. Went in to see his cardiologist. Okay, found out he had cardiac problems too. Guess what? His ejection fraction went from 35%, which is what it's been for 6 years, up to 45%. So the body now, as long as I stabilize Parkinson's, now I'm working on other things that need to be repaired. So the body dictates where these cells go. We don't have any choice. Once we put them in, all bets are off unless we actually stick them where we want them to go. The body takes over and just takes them where it wants to go and repairs what it wants to repair. Okay? So that's that sort of thing. Now, if you don't mind an HLA mismatch, in other words, if we take you and try to train, well, that's even more. Let's try 2 girls, because gender's not going to work anyway. So try a white girl and a black girl. That's fine. That's a nice allogeneic. If you don't mind that, perfectly fine. Not a problem. But if you want an HLA match, you have to do therapeutic cloning. It takes about a year to do therapeutic cloning, and another year to do down to the single cell type. So it takes you 2 years to get cells ready for transplant, plus $2 million. If you want autologous, I can get you stem cells in 2 days. 2 days, 2 years, a little bit different. So these are an identical match. So if you have a bone marrow, okay, if we bank your cells now, you have a bone marrow disease down the line where you need an identical match, so you have a twin sister. Yeah, you have a twin sister. Okay, if we banked your cells 2 years ago, you would have a twin sister sitting in a bottle,
Wow.
ready for transplant. Because we can form any cell type out of these cells, and we've done bone marrow transplants before with these cells. In animals, not in humans, in animals, but it does work. Okay, so, and I just, because my research runs on a shoestring, like I said, I'll take anybody into my research lab. And you walk by, grab you, put you to work, and this sort of thing. So I have a lot of collaborators. So anyway, that's the end of my talk. Do you have any questions?